Our next speaker is Thomas Lemberger, who's the editor of Molecular Systems Biology, published by NPG and the European Molecular Biology Organization. So think data, think computation, and his talk is titled Source Data and Other Article Enhancements. Thomas.
I don't know. Is the microphone working? Can you hear me? Okay. Okay. Thank you very much. It's a great pleasure to be here. So I will summarize this project. We have source data at EMBO. For those who are not very familiar, EMBO is the European Molecular Biology Organization. It sits in Heidelberg in Germany, and I'm the deputy head of scientific publication. So I have to say, to have a little bit of a disclaimer, because of course the audience is very broad today. I'm going to have a pretty biased view on what I'm going to present. It is really restricted to the life sciences and even more so to molecular life sciences. This is not at all to say that the project is restricted in its concept to these fields, but of course this is the way we are thinking. And now we are regrouping the 4 journals we are publishing under the umbrella of EMBO Press in partnership with Highwire and Wiley. So these are projects we are trying to develop in collaboration. So before I start, just to set up a little bit the stage, I'd like to start with this definition of what scientific publishing is. And I'd like to cite this book from the National Academies when they were trying to think already, I think 10 years ago, about why we need to share in science materials and data. And essentially, the publication is really dedicated to move science forward and is an exchange between the credit that the authors receive and the disclosure of scientific findings. So these are really sort of the 3 fundamental pillars, I think, that make scientific publishing survive. And the ultimate goal is, of course, to move science forward. This is what we want, and this is our first priority. And this works, of course, by the motivation of scientists working very hard and receiving credit for their work when they publish it. But it requires, in exchange, something that is not that easy to do, which is to disclose the findings and to disclose them completely so that others can reproduce them. And this entails disclosing, of course, the data and providing the data that have been produced by the research work. This has become very important in life science, where there is a massive production of data within the last maybe 10 years, but also the realization that science benefits from being more and more open, and making this data freely and openly available is actually at the core of the scientific process. There is also a growing concern about the issue of data integrity. We have to check that the data that have been produced are indeed real data and have not been falsified or altered. Now, this debate about data in scientific publishing is dominated, as everybody already mentioned it, by these keywords of big data. In life science, it has a very precise connotation because there has been a technological transformation in life sciences, with technologies able to produce very large amount of data, sequencing all the omics, so-called large-scale biology fields. So this has led to the creation of several public resources, public repositories, where different data types in life science can be deposited in a structured format. And these resources are invaluable because they are the only way, in a way, for the life science community to survive and to handle those data. And very early on, whether structural biologists or people involved in genomic biology, they had to organize their data, they had to come up with standards, and they created these large repositories. Now, it has created a situation or an ecosystem where scientific publishing papers and public repositories are living in somehow parallel worlds that are only loosely connected. And even our wording is kind of showing that we publish paper, but we deposit data sets. We don't publish data. Now, and this was very nicely summarized by a recent report. I'm sure most of you know that science is an open enterprise from the Royal Society. That the two vital components of the scientific endeavor, the idea, the concepts, or the demonstration, and the evidence, the data, are too frequently separated, and they live their independent lives. They have different systems of credits and of publication. Now, this domination by big data should not make us forget that, at least in life science, I'm not going to talk about physics or other disciplines, but in life science, journals actually publish data. There is no scientific journal that doesn't publish data. And there is this new now term coined data journals. All the biological, all the life science journals are actually data journals, and the data is published in the journal. So the sort of the vast majority of data published in life science is actually not large scale at all. It's very small scale, and it's just available in the papers. Now, this is important to realize because publishing in science is very important. It is the major channel to communicate peer-reviewed and quality-checked data. It plays, of course, a central role in the scientific careers of people because the scientific record is essentially the publications that are produced by a scientist. Yet we are facing enormous challenges. The rate of publishing is increasing. Of course, we are very pleased because we are publishers, but for the scientists, it is a problem because these papers, even though they are human readable, they are barely read by humans, and we depend on machine even though they are poorly machine readable. And this has serious consequences for search. If we want to search specific information, we rely on keyword-based methods, etc., but it becomes more and more difficult to have quick access to specific pieces of information and certainly to search for specific data sets. Now, this has, again, big consequences, the lack of availability of data to check whether they are reproducible, to really understand how they have been obtained, leads to potential mistrust in how science is working. And I'm sure you have seen this cover page in The Economist, how science goes wrong. It is not only linked to data, but also to the documentation, of course, of the methods and the protocols, which are kind of linked issues. So at EMBO Press, with our 4 journals, we sort of have this image of what is the paper, which is a layered structure that offers access to the information at several levels of granularity. For very general readers, we just provide the titles or the abstract, allowing to skim through, to really the main papers when people really want to read the study, and even an expanded version of the paper that contains all what was used to called supplementary information. But finally, the bottom layer here are the data sets, the computer code that really provide the foundational layer to a paper. And this is really the access to this foundational layer that we would like to promote and to facilitate to promote discoveries. Now, papers are constituted of a lot of text. We're essentially swimming in an ocean of text, which is fantastic for text mining. And we have heard wonderful demonstrations here of what advanced text mining and processing or language processing can achieve. And certainly in life science, text mining methods to identify biological entities, genes, proteins, the chemicals, are getting extremely good, and I think we are going to hear some additional talks today. Yet, to extract causality and relationships and directional relationships remains a challenge. I'm not saying it's impossible, it remains a challenge. And so this is a current, I think, a fair limitation for methods that depend only on automated text analysis. Now, papers are not only text, but they are also the figures. And in life science, essentially, this is really the core of a paper. We display the scientific data in figures, in images, in illustrations, curves, graphs, pictures, photographs. And essentially, the way we publish that is we convert the scientific result, which is highly structured, into a collection of pixels that are nice to see. They are nice to review because they are human readable, but they are not nice to reanalyze, and they are certainly even worse to search. And this is a problem that affects all the journals, whether they're open access or not. And essentially, the real data, the core of the paper, remains totally locked inside the paper because of that. So it is to to address this issue, this very large issue, of course, that we started the source data project at EMBO to think of and to develop tools that would allow us to publish figures as structured digital objects that link, on one hand, the human-readable version of the data, which is very important. It is important. In the end, the readers and the scientists have to understand the data. The computers will not really understand the data. But to link this human-readable illustration with machine-readable metadata that explain the data, and the source data, we call that the source data, which are the actual measurements and the data tables behind a figure. And this is to improve data transparency so that one can really see what is the real data that is behind a figure and not only the summary or the visual illustration. To make published data more usable by really explaining what is the content of this data and presenting them in a form that can allow some form of reuse. And finally, to enable data, what we call data-oriented search, and maybe I will come back to that. So the first dimension of this project, of course, is to provide the data in the first place and to really integrate that inside the papers. And this was, in a way, easy to implement, but it was an unknown how our community would really react to that. So since a couple of years now, we ask our authors when they produce quantitative data that are really important to develop a computer model of cells or biological processes. We ask not only to provide these beautiful pictures that shows the curves, but we actually ask to have the tables with the measurements. We extended that to categories of images. For those of you who are not biologists, it doesn't really matter, but we used to present little snippets of images here, which is sort of showing the essence of the data. But there can be a lot of background, there can be a lot of other things that can go right or wrong, and it is important actually not to only to show this very tiny window onto the data, but to show the full extent of the data, to show its quality, to show maybe additional signals that can be understood only later. And the good news is that we do this on a non-mandatory fashion. We just provide this possibility to authors to essentially archive their data with their own paper, which is very useful when the postdoc or the PhD is leaving for Siberia, the hard drive has been destroyed, and so on. And we have a very high degree of compliance, more than 50%, on a purely voluntary basis. So this really shows that for the scientific community, there is an internal motivation and there is a good understanding that it is in their interest to save the data that is behind the figures. And as I said, this has several purposes. First is data archival. For the authors, it's really a service to have the data sort of be ready for download in their own paper. Again, if most of the life science journals laboratories have rather poor data management, internal data management policies, so this is very, very useful. Data transparency so that readers can see more in detail what is behind an illustration. Data reuse, this plays a big role in systems biology where data integration, reanalysis of quantitative data is at the core of this type of science. And finally, data-oriented search. Now, providing the data is also putting a little pressure on the authors to really show what they have obtained. And this is important with regard to data integrity. Here I show, again, if you are not biologists, it doesn't matter. These pictures are beautiful. These are cells, microscopic images visualized by fluorescence, and they look actually very beautiful. However, we have a data integrity check in our journals, and we do that systematically. When we check how these images were actually obtained, you probably, even if you are not a biologist, you can see that there is some serious copy-pasting here and assembling and deleting of backgrounds to make those images look nice. So it is important to provide the data beyond what is just illustrated and made nice. To promote and to reduce the legal barriers to data reuse, we do essentially what has been done since many years in biology, which is to put the data in the public domain, and this is kind of the standard a procedure for most of the public databases. So all our data sets and source data are now explicitly under a CC0 waiver. It remains a question in practice what type of data we ask. We would like to have actionable data. We don't obligatorily request raw data that can be very difficult to reanalyze. And this is an example of one paper we published. This is the illustration. It shows a typical systems biology network of a complex process. This has been derived from an analysis of numerical data to see correlation profiles of features, cellular features. And this eventually stems from a high-content imaging screen that is something like 1 or 2 terabytes of imaging data. So, of course, we could ask for this really raw data so that the entire analysis can be regenerated. However, going from images to extracted features like the position of the nucleus in a cell, the type of cell, is very difficult to do and it's very specialized. And in a way, this intermediate layer for pre-processed data is more actionable than the raw data. And we let the community decide what is the most useful form of pre-processing of the data. Now, the second part, as I said, is to provide descriptive metadata that explain the content of the data and importantly, its structure. And this is something we totally lose in the way we publish data. In science, we are really lucky because we start from structured information. An experiment has been designed, has a structure. And we publish this as images, and we describe that in English text. And essentially, we denature all this structure after the fact. So we focus on the biological content of the data, and we scratch our head for a really long time how to do this, because, of course, in biology, like in other fields, there is a huge heterogeneity in terms of data types. There are no real standards for these small-scale experiments. And how can we find a common theme to encode sort of what is really important to encode? And when we talk about metadata, we talk less about technical or methodological metadata than really the metadata or the description of the design of an experiment. And the way we try to do this is, first of all, to have a sort of object-oriented way to describe an experiment, which is simply to list the biological components that are key part of an experiment. And in biology, this is very easy to do, and it is very natural to do, and the authors do this very, very clearly in their figure legends. There are also excellent resources that exist that allow to link terms and English words to very well-defined entries in those resources. The second level is maybe the most interesting and the most maybe profound, if I can use this word. It can be understood in a data set like this, where there was a measurement performed, measuring something, a component, a protein Y, as a function of different experimental conditions, plus or minus a drug that we will add to cells in culture, maybe with one gene that is missing or that is added, and the measurement showed the difference to these different perturbations. So there is really a causal link between controlled experimentations, experimental conditions, controlled perturbations, and the resulting measurement. And this is really the key. This is the reason why a scientist is doing an experiment in empirical science, is really to have a cause and effect that is a test that is well controlled and that will lead the scientist to deduce the mechanisms of how the process really works. So this is not a detail. Causality is really at the heart of many empirical science experiments. And by capturing this causality, the components that have been perturbed and the components that have been measured, we really have a good feeling now of why this experiment has been done or what is the causal test behind it, totally independently of the data type. It can be imaging, it can be graphs, it can be this type of evidence. And we have this sort of core now representation of an experiment that really represent the causal directional relationship between a component that has been altered in a controlled way and the resulting measurement and the assay that has been used. So we can apply this now across many types, not all, but many types of small-scale type of experimentation in biology. And finally, we can transform that in a machine-readable way. Again, the biological entities and chemical entities can be represented in an unambiguous way using existing resources. So we are developing the tools that allow to do that. We don't want to generate this information from scratch. We base our analysis on the legends to the figures. And again, the good news is that this way of representing an experiment is very natural to the scientists. And we didn't invent it, of course. We just read the figure legends, and this is what emerged, that people just describe what they have measured, what they have perturbed, what is the assay, and it is in the way very easy to capture this information simply by tagging the natural text that has been generated by the authors. Since this text is very, very small, we could do it in an automated fashion, but it almost doesn't really provide any advantage, because for a trained reader, this is almost no effort to locate. The difficult bit is to identify the causality. Maybe it can be done in an automated way. We don't know. At the moment, we need really somebody who understands the purpose of the experiment, sort of a trained curator, who can isolate what was on the side of the perturbation, what is the cause, and what is the effect. And sometimes it's actually not that easy. We tag those terms. We can import it in a database, transform that into machine-readable identifiers, and express a little bit of ambiguity when necessary. And we are now in the process of developing workflows with EmboPress in collaboration with our publishers to not only verify the integrity of the data, but not only to verify the proper data presentation in the illustration, but also to have a step of semantic enrichment and structuring of the metadata to capture this metadata that are around figures. So what is it good for? Well, it is, of course, good to describe in a rigorous way an experiment that is published. If somebody forgets to say what is the assay that was used to produce a figure, if somebody forgets what has been actually measured, it is not really rigorous science. So it just de facto imposes a standard that is a sort of minimal information to include in a figure. But it is also useful for search. We capture causality. We have directional links, and directional links that are really linked to the way scientists look at the biological objects. So the idea of an implementation, for example, at the article level, would be that there would be a query, like a figure that is of interest for the readers or for the person doing a search. And it should be possible to retrieve from the annotated papers figures that resemble this one, that address a similar question with the same causality, whether this drug is influencing this protein, and even to do this in a sort of a fuzzy way, to gather the images or the experiments that are really related in terms of their purpose to the query. And this way we asked our readers whether this would be an interesting feature. Maybe there is no surprise. There is a quite robust support for having this type of things. Actually, people think it's not possible to do. So this leads me to the concept of data-oriented search. If we use PubMed and keyword-based search, which is what scientists sadly use, 90% of them use PubMed for their search. If we ask a simple question, and this is a biological question, you don't have to worry what it means, but finding the substrate of a kinase, CDK1 kinase, this was a question suggested to me by Tim Hunt, who is Nobel Prize for having discovered CDK1. And he said, well, this is really bloody difficult to find all the papers. I should know, but I tried to do that, and it's really difficult. And you end up writing very baroque queries, with lots of Boolean association, try to capture whatever you can. You find hundreds of papers, you have to open them all. And the only thing you would like to find is the experiment that shows, that CDK1 was affected in some way, and another protein or another gene has changed. So this is difficult with keywords. It's maybe not impossible, but it is difficult. And can you see that? Yes. So now, just as a demonstration, as a toy demonstration, I I'll show you what could be possible if we can isolate the components in the experiments and if we can really separate them in cause and effect. And we could find experiments where CDK1, this protein, has been perturbed, has been altered in a controlled way, and find all the experiments where there's some effects have been measured. And in this way, we would retrieve now the experimental evidence, where every time CDK1, this protein, the little bubble here is a protein, has been altered and something else has been observed, and have direct access to the specific papers and, of course, read the papers. So have access to the papers from the data instead of having access to the data from the papers. And importantly, the causality here is really important, because if you would now ask for the reverse, for experiments where CDK1 was measured, do I have a missing? The results, of course, would be different. And in this case, you would find experiments that shows regulators of CDK1, something that is upstream of this protein. So again, a totally different set of papers depending on the causality. Having found one of these papers, one can read the paper, focus on one piece of data, and this is the second implementation, sort of transparently to the user, to find related data sets that resemble this experiment, but also related data sets that are either downstream or upstream in the chain of causality, navigate away from the paper to find what is a regulator of CDK1 and go from there to the paper. And in a couple of clicks, go really through the actual biology and find the papers depending on the connections between the experiments. So I think it doesn't take too much imagination to see that a system like this will allow to combine data sets across papers to generate new hypotheses and to navigate this network of connected experiments. So the future of publishing, we don't really know what it's going to be. Maybe it's going to be like this, you know. But we think that the human readable version of the paper is essential and will stay for a very long time, maybe not as a PDF, but humans need to understand the science and to analyze them and to have their intelligence analyzing the data. But behind this human version, there will be a machine-readable version that contains the data and contains an explanation of the data to increase global data mining and generate a new discovery. And this is really what we are going to focus, one of our main focus with EMBO Press. Thank you very much.
Thank you, Thomas. There's time for maybe one question while we're swapping out computers.
Anthony Watkinson, Cyber Research. I think everybody would agree that what you're doing is fantastic and wonderful, okay? But it costs. It costs a lot of human intervention. You must spend a great deal of money on this. Elsevier spend a great deal of money. Other people, Nature Publishing Group, what about the smaller people being eased out so that it could be, in the end, just a few publishers able to do the stuff which is best for the researcher?
Yeah, I think it's a really good question. Frankly, it's an open question. It costs a lot of money. We try precisely to focus on the figure on small section of the papers in a sort of very pragmatic way to minimize the cost. And we are fairly optimistic now that we can really do this. And this is why I mentioned the workflows. We try to save time on other places that are maybe less important to the scientists. We have been spending a lot of money doing typesetting, doing layout, doing these kind of things to check, whether it's the UK or the US English spelling, totally ridiculous things that are of no relevance whatsoever for scientists. And so we really try to see where we can save time and money in that to free something like 10% to 15% additional resources. And we think this is a ballpark figure we need to do these kind of things. Is essentially, it corresponds to 10 to 15% additional work that has to be saved somewhere else, or that has to be paid for by authors. And of course, with the funding agencies now that have strong mandates for data deposition, it is important, and this was one of the slides I skipped, I'm sorry. It is important to realize that scientific publishing is really a core part of the research infrastructure. And it's very good to put data in repositories that are funded, but it is also very good to put the data into the papers. And this costs money, but it is really in the interest of the general scientific community.
Okay, thank you again, Thomas.